Is Group 33, whose members are Wachira Kevin, Otieno Tilen, Kevin Onditi, Abraham Kiprop, and I, Gorevan Otieno. Our topic of discussion will be the description of osmoregulatory organs of invertebrates. In this discussion, we are going to cover its definition of osmoregulation, importance, how it works, and examples in invertebrates.
Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on invertebrate osmoregulators. Now, in invertebrates, specialized excretory tubules are present, and this specialized excretory tubules they participate in osmoregulation and they also involve in the excretion process. Now, in invertebrates, for osmoregulation and for excretion, specialized tubules are present. Now, in invertebrates, three common types of this specialized tubules are: one is protonephridia, which is found in flatworms and the larva of mollusk; metanephridia, found in annelids, for example, the earthworm; and in adult mollusk, metanephridia is present, whereas the Malpighian tubules are found in insect and other arthropod. Now, all this specialized tubule, the mechanism for osmoregulation and excretion is different in all this invertebrates. So now, first we are discussing about the protonephridia. Now, the flatworm Dugacia, also known as planaria, provides us an example of the simplest form of invertebrate excretory tubule, which is known as the protonephridium. Now, in the body of Dugacia, two branching network of protonephridia are present, and these two branching network of protonephridia they run to the entire length of the body. In the figure, let me show you this. This is the body of Dugacia, and in the entire length of the body, as you can see, this one and this one. Okay, these two are the protonephridia. Now, the proximal branch of the tubular network end with the phlegm cell. So, at the proximal end, phlegm cells are present in this tubular network, and the phlegm cells are the cells where the filtration takes place. So, here you can see the in the structure of protonephridia, this part network of tubule, and this portion they have zoom. So here on the proximal end, as you can see, this is the phlegm cell is present, and this is also all are phlegm cell, and this phlegm cell, as you can see, they are surrounded by hemolymph. So the phlegm cell are the cell where the filtration process is occurring. Now, in each phlegm cell, a bundle of cilia is present, and this cilia bundle extend into the tubule, and the continuous beating of cilia produces a current. Okay, so the fluid, okay, from the hemolymph drawn in the tubule. So here you can see, here also. Okay, these are the cilia and they are continuous beating. So that's why what is happening, this is the hemolymph and the fluid is drawn in the phlegm cell. Now the fluid passes into this tubule. Okay, so now filtrate fluid from hemolymph has now entered into the tubule. Okay, so hemolymph, see what is hemolymph? Hemolymph in the invertebrate is considered equivalent to blood, which is present around this protonephridia or the phlegm cell of the protonephridia, or around the all tubules of the protonephridia. This hemolymph is present. So now we have seen that in the phlegm cell, the filtration process take place, and this filtrate pass through the tubules of protonephridia. So when the fluid passes in the tubular network, what happens? Some molecule and ions are reabsorbed and they again enter into the hemolymph. While the nitrogenous waste which are present in the filtrate, they are not going to reabsorb, and the tubule are going to secrete them outside the body. So now the filtrate which is having nitrogenous waste is known as the urine, and this urine is excreted through the pores, and these pores are present on the distal end of the tubule and they open on the body surface. So simply see what exactly the mechanism is. This is the hemolymph. Now hemolymph fluid from the hemolymph enter. So the filtrate, here the filtrate is formed. Okay. Now where this filtrate is going? So for example, this one, this one, this one. So this is the filtrate, this is the filtrate is coming. Okay, and this is the tubule. This is the tubule. Now this cell which are present in the tubule, they are going to reabsorb some important solutes and ions, and the nitrogenous waste, okay, it is going to remove by this pore. Okay, so this pore or the opening opens, okay, and it is present on the distal end of the tubule, and this pore opens on the body surface of the Dugacia, and the urine is released. Okay, so this is how the urine is formed, and this is how the excretion take place in Dugacia with the help of protonephridia. And I'm showing you the entire figure. This one. Now we are discussing about the metanephridia. Okay. See, in most annelids, such as earthworm, having metanephridia. Metanephridia in earthworm is an excretory organ, and this metanephridia collect fluid. It receives fluid directly from the xylem. Okay. Now xylem is the body cavity in which all the organs are present. Okay, and from this xylemic fluid, the metanephridia receives the fluid. It collects the fluid. Now, how many metanephridias are there? So, see, a pair of metanephridia, that means two nephridia, are present in each segment of an annelid, of an earthworm. And this metanephridia, they are immersed in coelomic fluid. They are deep in the coelomic fluid, and they are covered on their surface, on the metanephridia, on their surface, envelope means covering of blood capillary network is present. Okay. Then the initial part of metanephridia, where the opening is present, internal opening, so that initial portion is known as nephrostome. And inside the nephrostome, cilias are present. So when the coelomic fluid enters into nephrostome, so as the cilias are beating, so the fluid is filtrate and it enters into the metanephridia. Okay. So as the cilias are beating, cilias are drawing the fluid from coelomic fluid, and this coelomic fluid enters into the collecting tubule of metanephridia. Then this fluid passes to the bladder. Okay. And finally, the fluid will be expelled out through the opening which is present on the body surface. But before that, a reabsorption process is going to take place. So first, we are looking at this structure of metanephridia. Let me show you the metanephridia first. See, this is the segment. This is the segment of earthworm, and here this entire structure is the metanephridia. Okay, this initial portion of metanephridia is known as nephrostome, and as you can see in the nephrostome, this internal opening is present. Now, when we look inside the nephrostome, see, this is the opening, and this is the nephrostome, and inside the cilias are present, and these cilias are beating. So due to this beating of cilia, the fluid from the coelom enters into this opening. Fluid is drawn inside the nephrostome. Okay. Then the fluid is entering into the fluid pass to this one is the collecting duct. Okay. So the fluid travel through the collecting duct, then the fluid finally going to enter into the bladder, and here in the bladder it is going to store for some time. Now, before that, reabsorption process take place. So here, as you can see, this is the capillary network on the metanephridia collecting tubule. Okay, so some solutes, some ions are reabsorbed and they enter into this capillary, and the remaining filtrate fluid enters into the bladder. Here it is stored, and then from the bladder with this duct, and this duct is opening outside. So this way the urine is excreted outside. Okay, through external opening. Now, one more figure I would like to show you. For example, over here, okay, here you can see all the segment of the earthworm. and here they have shown one segment, and in the one segment, this green in color, okay, is the meta nephridia. Now in the nephrostome, hemolymph or the coelomic fluid, because annelids, okay, phylum. So see, coelomic fluid enters into this nephrostome, okay, due to the beating of cilia. Okay. This is the first step. Then in the second step, reabsorption in convoluted section of tubule. Okay. After reabsorption, finally urine enters into the bladder. Here it is stored for a while, and once the bladder is filled with the urine, so the urine is released outside from the spore. Okay, so these are the steps by which the urine formation take place in the metanephridium of an earthworm. Okay. Okay. Now first, see, as we know, earthworm, they live inside the damp soil, and therefore usually they experience a net uptake of water by osmosis. So from this damp soil, what happens? Water by osmosis process enters into the earthworm's body. First, water crosses the skin and enters in the body of earthworm. Now the metanephridia of earthworm balances the water influx by producing urine that is dilute. Metanephridia produces the urine which is hypoosmotic to body fluid. Now, in producing hypoosmotic filtrate, the transport epithelium, which is present in the wall of convoluted tubule, absorbs solute, and this solute in some ions returns into the blood of capillary network, and this blood capillary, they are enveloping the convoluted tubule of metanephridia. So we can say filtrate, the solutes and ions, they are reabsorbed and they are entering into the blood. Now the rest of the urine, rest of the filtrate is now known as urine, and this urine moves forward. It is stored in the bladder, and then it is excreted out by the pore which is present on the body surface, on the body wall. Okay. So here we can say the metanephridia of an earthworm serves as both the excitatory and the osmoregulatory function. Okay. Because the it is excitatory. Metanephridia is excitatory because it is producing urine, it is excreting urine. And second thing, it is also responsible for the balance of water in the body. Okay. So that's why it is both the function, the metanephridia is function of excretion, and osmoregulation is performed by metanephridia. Now we are discussing the Malpighian tubule. Malpighian tubules are the excretory tubule of insect. Now, in the Malpighian tubule, the proximal end is closed, and this Malpighian tubule are immersed in the hemolymph. So here, see, the insect body is shown. All the Malpighian tubule. This is the hemolymph they are showing, and these are all Malpighian tubule. Okay. So this end, proximal end, is closed and distal end open into the gut. So proximal end, which is closed one, immersed in the hemolymph, and distal end open into the gut. Now, unlike the excretory system, Malpighian tubule do not use pressure filtration step. In urine formation, first step we know is the pressure filtration, but that step is absent in the insect. So instead, what happens in the insect? See, first from hemolymph, Ka plus enters into the lumen of proximal segment of Malpighian tubule, and the entry of K plus, due to that, the tubular fluid becomes positively charged, and as the Malpighian tubule fluid, due to the presence of K plus, it is positively charged, so it is followed by the entry of Cl minus from the hemolymph. Okay, so from the hemolymph, what is happening? First, K plus is entering. Okay, then entry of K plus as the positive charge develop in the Malpighian tubule fluid. From hemolymph, Cl minus enters. Now, as the K plus and Cl minus are present, so now water enters from hemolymph. Now K plus, Cl minus and water enter into hemolymph. So now from hemolymph, organic waste, for example, nitrogenous waste, uric acid, enters into the uric acid, organic waste enters into this Malpighian tubule. So there are one is K plus is there in Malpighian tubule, second is Cl minus is there, then water is there, and now organic waste, nitrogenous waste, uric acid is there. Okay, and all this substances, they enter into the gut. Okay, and all the substance, they move along the gut. Finally, they reach by passing hindgut. Finally, they reach into the rectum, and the wall of, as you can see, the wall of hindgut and rectum, the cell, they are reabsorbing some of the substance. So K plus reabsorb, Cl minus reabsorb, water also reabsorb, but uric acid is not absorbed. So as uric acid is present over here, uric acid precipitates as a crystal. So now here, this is the GI tract. We know digestion process, and after once the digestion process is completed, the fecus is formed. So here the fecus mixes with the uric acid, and this fecus and uric acid expel out through the anus outside the body. So this is about the Malpighian tubules. Okay. Now we are discussing about the green gland or the antennal gland. Okay, see the anatomy of crayfish. Okay, and here you can see green gland. Here coxal gland, the parts of the coxal gland. Here in this figure also see crustacea. Okay, see, and this is the dissected crayfish. And see the second antenna. At the base of second antenna, you can see the opening of green gland, and this is the entire green gland. So the green gland consists of three basic parts. One is end sac, second is convoluted tubule, and exit duct. And in this figure, internal anatomy of female crayfish. See, okay. Green gland, and you see the opening of green gland. So the opening of green gland at the base of second antenna. Antennal gland or the green gland of crayfish. These are the parts of green gland: end sac, labyrinth, tubule, bladder, duct. Now in this figure also, okay, antennal gland of crayfish is shown. So the first part is the xylem sac, also known as end sac. Now second portion, labyrinth, followed by nephridial canal. Nephridial canal open into bladder. Then this part, ureter, and ureter opens outside. Okay. So see, in most crustacean, for example crayfish and prawn, have a pair of specialized excretory structures known as antennal gland or green gland. So antennal gland or the green glands are the excretory structure which are present in most of the crustacean. For example, in the crayfish and prawn. And where they are present, okay, so they are present at the base of second antenna. Now what the green gland is made up of, the component of green gland. So each antennal gland or the green gland comprise of a bulbous end sac, also known as xylem sac. So first the xylem sac present. It is followed by labyrinth. Xylem sac followed by labyrinth. labyrinth followed by narrow tubular nephridial canal, labyrinth open into nephridial canal, and this nephridial canal open into bladder. Okay. Now let's see how the urine is formed in the green gland. So this coelomic sac, the initial part of green gland, this coelom sac is immersed in blood lacuna. Okay, so here let me draw one blood lacuna. So this is the blood lacuna. Okay, and in the blood lacuna, blood is there. So this coelomic sac is surrounded by blood lacuna. Now what happens? Filtration of blood take place at coelom sac. Okay, blood is filtered at coelom sac. So this coelom sac produces the filtrate or the excretory fluid, and this excretory fluid or filtrate now passes through the labyrinth and also from this nephridial canal. Now in the wall of nephridial canal and the labyrinth, the cells are involved in the reabsorption process, that is the selective reabsorption. Okay, so some solutes and ions, they are coming out from the labyrinth and nephridia. Okay, and the remaining filtered fluid or the excretory fluid, which is now converted into urine, that urine passes along with this nephridial canal, and finally the urine enters into the bladder where the urine is stored. Okay, then the urine enters into the ureter. This is the ureter, and ureters open out by a pore, and this pore is present at the base of the second antenna. Okay, so this is how this crustacean, they are excreting urine, they are forming the urine with the help of green gland or the antennal gland. Now I would like to show you all the figure, but before that let's read out this information regarding the antennal gland or the green gland. See, either of a pair of duct, coelom duct, found in the third segment of crustacea and opening to the exterior at the base of second antenna. Now the green gland or the antennal gland, they function as osmoregulatory organ. For example, the antennal gland of freshwater crayfish, Astacus, fluid is filtered from the blood into end sac, also known as coelom sac, and this fluid passes through the tubular labyrinth. And here in tubular labyrinth, ions and some solutes are reabsorbed. And once the ions and solutes are reabsorbed, this filtrate fluid is now known as hypotonic urine. This urine passed through renal tubule, also known as nephridial canal, and now urine enters into the bladder. And now from bladder, urine enters into the ureter, and by a pore which is present at the base of second antenna, the urine is expelled out, urine is excreted outside the body. So one by one, I would like to show you all the figures. This we have seen, okay, we have discussed this. This we have seen. Protonephridia and planaria. With this step, you can understand the entire mechanism of urine formation. Okay. You can see the cap cell and tube cell together. So this is the phlegm bulb. Okay. Phlegm bulb, also known as phlegm cell. And inside you can see the cilia are present. See the interstitial fluid filtrate, produce, travel into tubule and opening in the body wall. Protonephridia and planaria. And in this figure, metanephridia of an earthworm, and right hand side, Malpighian tubules of insects. Now here, in this figure, the protonephridia of flatworms, phlegm cell, fluid, cilia, collecting tubule, excretory pore, the nephridia of annelids, and this way, the Malpighian tubule of insects, here in this tubule of protonephridia. Okay, see the phlegm cell, how the cilia are beating, cilia drawing interstitial fluid, okay. Reabsorption, then urine expel out. So this is the opening in body wall. And in this figure, okay, components of metanephridium. So internal opening which is present in nephrostome, then collecting tubule, bladder is there, capillary network present on the collecting tubule, and you can see the external opening. Excretory organ of an insect, Malpighian tubules. Then in this figure, okay, protonephridial filtration system of flatworms. Okay. Step by step you can understand the entire mechanism 1, 2, 3. And this is for earthworm, right hand side metanephridia filtration system of annelids. Malpighian tubules from the secretory system of an insect. 1, 2, 3. Okay. So with this, we have completed this lecture. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Koshti sir. I am from Ahmedabad, India. Bye bye. Namaste.
From our discussion of osmoregulation in invertebrate, I can conclude our discussion by saying osmoregulation is essential for invertebrates to maintain water-ion balance, ensuring survival in diverse environments. We explore the key osmoregulation organs like the protonidia and metanephridia and Malpighian tubules. Each adapt to specific and specific needs. This process helps in water removal, energy conservation, and adaptations, eligibility invertebrates to thrive in fresh marine and terrestrial habitat. As environmental conditions change, understanding osmoregulation becomes even more important. Thank you for your time. I am happy to take any